People from the waiting room, sorry.
Yep, that's good, that's great. So I'm going to leave, Anita will be introducing our wonderful speakers. So I'm going to start off with acknowledgement of country. So I'm presenting to you on the lands, sorry, I am here on the lands of the Bunurong people of the Kulin Nation, and I wish to acknowledge them as traditional owners. And I would also like to pay my respects to their elders past and present, and Aboriginal elders of other communities who may be here today. And of course, many of you will be on various lands of Indigenous people, and we acknowledge the lands that all of us are on. So it gives us great pleasure to present this second seminar in our series of seminars to do with the Future Food Hallmark Research Institute, of which I'm the chair, and Dr. Minha is the academic convener, and Dr. Alita Lawrence is one of the leaders of one of the projects. So our project is on future food, and of course future food is so important and pivotal for us in terms of going forward. So our project certainly investigates sustainability aspects, nutrition and health, which we're very fortunate to have Roger and Peter today, policy and social drivers, regulation, how to produce these alternative proteins, and also the blending of proteins. So if you're interested in this project and want to engage in it, please do put the link for it into the chat function. So please do go to the chat function and press that link. And now I'll hand over to Anita to do the introductions. Thank you, Anita.
Well, good morning, everyone. And it's wonderful that we can join across the world for today's webinar. So I would also like to acknowledge the elders and descendants of the people on the land on which I'm working, which, like Robin, is the Bunurong people of the Kulin Nation. And I do pay my respects as well to their elders, past, present, and emerging. So today I'm absolutely delighted that we have Roger Clements and Peter Presman speaking to us today, all the way from the US. They tell me it's afternoon for them, so the time zones have worked out quite well. It's not in the middle of the night for anyone. And Peter and Roger are going to talk about the scientific, medical, and regulatory landscape of alternative protein sources and their impact on food allergies and sustainability. So welcome to Melbourne, or virtual Melbourne, Roger and Peter. We used to be the world's most livable city. Now we're the world's most locked down, but today is a very exciting day because as well as you speaking to us, it's the first day of almost normality for us after very, very long lockdowns. So we're very pleased that you can come and talk to us today. Before you start, I'd like to introduce you a little bit. Both Roger and Peter are associated with Polyscience Consulting in Chatsworth, California. And Roger is an adjunct professor of pharmacology and pharmaceutical sciences. And he's also assistant professor of regulatory and quality sciences at the University of Southern California. For more than 20 years, Roger has been responsible for courses in food and drug toxicology, food science, and the regulation of foods and dietary supplements. Roger develops and instructs on graduate global nutrition courses within the University of Southern California School of Medicine, and also on a course on food science for lawyers at Michigan State University. Roger is an elected fellow of the American Society of Nutrition, the American College of Nutrition, the Institute of Food Technologists, and the International Academy of Food Science and Technology. He has also published more than 50 original manuscripts and commentaries. So welcome, Roger.
Thank you.
Peter Presman is the Director of Medical Operations at Polyscience Consulting in California, and also an Assistant Professor of Clinical Medicine at Sabre University College of Medicine on the island of Sabre. And I'm quite intrigued by this island of Sabre, I must admit. Peter has worked in both internal medicine and surgical education, focusing on nutritional support. He's also a reviewer for the Journal of Food Science, the Journal of Food and Chemical Toxicology, and he serves on the editorial board of Toxicology Research and Application. So welcome, Peter. So Roger and Peter have kindly agreed to present for about 30 to 40 minutes. And after that, there'll be about 20 minutes for questions. So you can either type your questions into the chat as we go along, and then I'll try and read them or give you a chance to ask them yourself in person at the end of the presentations. Or if not, at the end, you can put up your virtual hand. So do keep thinking of questions as we go along. So over to you, Roger and Peter.
Call from Evan Rouse.
Too many phone calls. I can't do this. Too many emergency phone calls. Okay, let me do a share screen. Thank you so much for the lovely and this great opportunity to be with you today. Peter, it's at 6 o'clock at night for him. So I'll do this. I'm going to share a screen, I think.
It just takes a moment to come, I think, Roger.
How's that?
Coming. And then you just need to do your one where you change the view, because we can see your notes. Yeah. Oh. So last time you just flipped it. What did you do? The trickiest part of this at the start, isn't it? Getting them projecting.
How's this?
It's coming. And then you did something last time where you flipped it so the presenter view was on this screen. I'm trying to think now, when you've projected, you just need to go to that little 3 dots, is it, where you change that, I think? Yep.
We just did slideshow from the beginning.
And then you go where the little dots are, you switch the, oh, that's the one, yeah, that's the one.
How's this?
Yes, that's the one, yeah.
Are we good?
That's it.
Perfect.
Perfect, Roger.
Thank you. I appreciate, I am technologically challenged, and I appreciate your patience. Dr. Preston and I really appreciate the opportunity to be with you today. Obviously, this is a very important topic internationally. Dr. Preston and I have served the underserved population of the world, and we still have a passion for doing just that. We could talk to you probably offline about that work that we're doing internationally. But today, to your excellent comments, Anita, we want to address this topic. We actually spoke on this topic in Poland a couple of weeks ago, and thanks to Min's excellent and kind remarks, we've updated this, and we're here today to share that with you. Obviously, this has become the new landscape to look at alternative proteins. This is just the myriad of folks that are involved with this. And our concern is from a clinical perspective and also allergy perspective. And even though sustainability is the buzzword of today, there are so many definitions of sustainability. I think it would be nice if that, in fact, that definition were harmonized. This is a recent graphic provided by some of our colleagues, and by the way, you can have all the slides. You just give us a URL, we'll send them to you, and so far you and your team can use these. You can see the areas that are in red or that are projected red there in Africa and South America, where perhaps the market isn't there. But where Dr. Preston and I have served both throughout South America and parts of Africa, and even parts of where you see where India is there here in the gold area, and to the right you see Malaysia, Indonesia, and that space. This is a very serious issue because we've actually worked with populations that are underserved, and part of the underserved parts that you see in parts of Africa, and particularly in Ghana, and likewise, in fact, the quality of protein leaves a lot to be desired. Adequate protein is a bit of a challenge in many of these population groups. And clearly, as you can see here, that if the world market is saying, well, what about mainstream? And I'd like to refer you to the last 2 bullets. It's really complex. It's really more than protein. And we need to understand how we measure protein and what are the physiological dynamics of proteins, and also some allergy issues might associate with these kinds of protein. So clearly there's some health implications across different population groups, and those population groups are very concerning for us. This is a spin-off of a paper actually that was published about 10 years or so ago in the UK, and now they've updated this paper, which is really quite good, on the future of food. And you see in the center there are a number of technology spaces that need to be addressed. I find it really interesting that when it comes to technology, the general consumers will accept technology in virtually every aspect of their lives, except when it comes to food. And yet Peter and I would say that technology in terms of the future of agriculture and providing food for the many of the world is really going to be critical. This is a survey that was done out of the UK, actually throughout Europe, of about almost 2,000 folks. So the consumers say, well, I can accept meat, dairy, and seafood. They've been part of the mainstay of their diets for many years. But also when it plants, well, okay, maybe. But when you're talking about insects and single cells and some type of biotechnology and fermentation technology, they're not quite so sure. So even that gives us an opportunity to educate the masses on what plant technology brings to us and what that really means to the health and well-being of the rest of the world. And insects, Dr. Pressman has some really great comments about insects, which we'll touch base with a little bit later. But we have to look at about not only allergies, but also perhaps viruses that are associated with different plants as well as insects. These are innate. But what does it mean?
Yeah, I'd like to, Raj, may I interrupt a little bit?
Oh, by all means,
Peter.
Peter. Yes.
Real quick, since we're looking at plant and insect here, I think we, especially now in this age of the pandemic, of all the COVIDization of science, it's appropriate to talk about an area that we believe is significantly underrepresented, both in research and in the commercial technology realm, and that's over. We know that phytoviruses are highly prevalent in plants worldwide, obviously including vegetables and fruits, and that humans, and in fact all animals, are exposed on a daily basis to these viruses, among which several are extremely stable. It is currently accepted, it has been the traditional view in evolutionary virology and in viral genetics that a strict separation exists between plant and vertebrate viruses regarding host range and pathogenicity, especially to humans and other vertebrates. However, there's reason to believe that, and very good scientific reason to believe, we feel that that plant viruses can pose significant pathogenicity, and that there are, in fact, many examples that we know of in the emerging literature in which phytoviruses circulate and propagate in insect vectors. So we really have to wonder now, especially when we see the popularity of plant-based dietary patterns being embraced so enthusiastically and so broadly, when we see insects as an alternative protein source being embraced, we have to be concerned, I think, about the evidence of immune responses that we are beginning to see to plant viruses in not only in non-human vertebrates, but in humans as well. Now, we know that hundreds of plant viruses have been sequenced and have been identified in the human GI tract. There is a real virome within the microbiome. We have long believed, as I said, that the entry of plant viruses into human genomes is quite another story. However, the evidence to date, as I've said, I think raises an unresolved issue, and there is, as I say, emerging data that really should prompt us to intensify, at the very least, our study of, our surveillance of phytoviruses and their genetic stability within animal and human hosts. And I think that becomes even more salient when we consider inocula, the size of the inoculum with plant-based diets, the size of the inoculum, the intensity of the exposure, particularly in insect-based products, which grind up 100 to 200 insects in some insect protein bars. You're looking at a concentrated densovirus inoculum, and we do not survey for these viruses. We don't know much about their genetic stability in human hosts. And we do know, for example, that densoviruses, which infect crickets widely and can be catastrophic within those populations, can mutate, we can observe genetic shift and drift in those viruses, as well as frank mutations. And the gain of function in those mutations, in other words, the virulence factors have definitely increased in insect populations. So we really have to think about this, and we've really got to do some further investigation. So I apologize for being polemical about this, but I think we've talked about it for years now, and I think we are obliged to talk about it much more seriously and do something about it, particularly in the face of what we have faced in the last 2 years and what we are facing.
Excellent comments, Peter. We have done some work in the space and grass documentation, and I can share some of that information with you. And you'll see some of the points that Dr. Preston brings up will be on the future of our slides. We see here that any luck at all, there we go. I think one of the areas is that, in fact, how do you measure protein in food? This gives you some definitions here from the United States perspective as from the FDA website. The reality is, you see the top one in particular, that in fact the definition, it says contains all essential amino acids. Well, no. In fact, all living creatures contain all the amino acids, but the proportions are critical and the digestibilities are different. And yet it is not until you get down to the second one, incomplete amino proteins are missing or not enough of. None of these definitions you'll see here talks about digestibility or bioavailability. We think that's another really important area for us to discuss. FAO talks about insects. Dr. Pressman and I have done some work in this space. Dr. Pressman will present some of his work, but it's becoming popular. Because of what we do, safety is paramount. And Aladdin raised some excellent point just a moment ago in the chat. If we look at how do we feed the growing population that's over 65, I'm well over that. And so is Peter. How about that? Peter's birthday is in a couple of weeks. He'll join the 70 crowd. But the safety is critical. And with all the panels that I've sat on for the decades, people talk about microbiology, but no one talks about viruses. Even when the Wuhan issue surfaced a couple of years ago, and I was working with one of my clients in the province there in China, I said, do you check for viruses? And the answer was no. I said, that's a mistake. And we're going to show you why that's a mistake. So Dr. Pressman, the protein insect industry is going large. If you want to copy the copy, you see here this, the latest issue. I think it's really interesting to do from a marketing perspective. But more importantly, from our perspective, we need to understand it from basic biology. And with
these guys will scream about
Dr. Preston, you have a comment?
I do very quickly. In a general sense, a lot of these budding companies, which are now growing far out of the cottage industry that they were only 5, 6, 7 years ago, to a major part of the market. This is no longer boutique, and they will scream very self-righteously about addressing the protein calorie needs of the underserved in the developing world and in resource poor environments and in the most austere settings. And they'll talk about the protein, the quantity of protein that is generated here in a very sustainable fashion. And yet, what these people are bound to is the traditional definition of protein, which has to do with nitrogen content. So you all are familiar with nitrogen content and the classical analysis of protein based on nitrogen content. The issue with insects, however, and crickets in particular, is that the chitin exoskeleton is essentially a tremendous source of non-protein nitrogen, so that the estimates of protein quantity and protein availability are significantly in excess of the reality. And then we have to consider, as Dr. Clemens has said, the proportion of essential amino acids. What are the rate limiting amino acids? And furthermore, what is the digestibility? What is the unique bioavailability here, which we can measure at the bench? Raj, would you like to elaborate a little bit?
You know, I've done that a great deal in my time right here, but we'll actually right a bit. You'll see here in terms of insects, they're ubiquitous. Dr. Preston has served the Navy, and this gives you a perspective that, in fact, as crickets grow, they produce a lot of quantity here. And we chose this as an example here. So this relates to 10 to the 4, that's 10,000 enterprises. That's the mass of insects that are available for us to consider for maybe a food supply.
So that's that's a great deal of aminopolysaccharide. So aminopolysaccharides are very interesting in terms of their biomedicine applications.
Yes, it
but
we'll
as far as a nutritional source goes, not very effective and not at all relevant.
And you see some of the biomedicine work that we actually, Dr. Preston did a few years ago. We'll share that with you. Obviously, insects are quite ubiquitous, so we won't relay this. So we'll go on to this area. It's interesting that the previous graphic indicated some bars. I actually made a presentation with Dr. Preston at a local meeting for a group of dietitians, and I said, so I gave out all of these cricket bars. I said, show me the safety data. Well, as they ravaged through the bars, I said, there aren't any safety data, none whatsoever. So I think we have to be very, very careful as we go on down this path. To Dr. Preston's excellent remark, we did some analysis a few years back. And you see here when it comes to crickets, in particular, limiting amino acids, anything below 1. So here we have the sulfur amino acids, the methionine and cysteine. Tryptophan is low, and obviously lysine is low, like we see typically in cereals. Then you make some little corrections for that, and the right-hand side of your screen, you'll see that, of course. So the amino acids way over there on the far right-hand side, when you correct for non-protein nitrogen, that Dr. Preston brought to the surface, you see that, in fact, now we have low threonine, we have low sulfur amino acids, we have low tryptophan, we have low histidine, we have low leucine, and low lysine. So clearly, and none of the protein that we see in the market today addresses this very important issue. It was 1996. The FDA said, well, non-protein nitrogen is not a serious issue. It's less than 2%. But as we move on to other alternative sources of protein, this becomes a serious issue when it comes to identifying opportunities, and importantly, identifying issues that could affect particularly malnourished individuals. So we would contend that, in fact, the classic Keladol method is inappropriate. With my friend Jeff Moore at the USP in Washington, D.C., published a paper 10 years ago, now 11 years ago, and tried to get the FDA to take some action on methodology. No action has been taken. And clearly, to Dr. Preston's excellent remark, non-proteinized should not even be considered the methodology today. So what is the appropriate international accepted method? It's still based, regardless of the method, PER, PDCAS, and DIAS, it still has the fundamental flaw. The fundamental flaw is about Keldol. So what do we do about that? Well, this is this you see in this graphic, this was from 1941 from the USDA. The data are true then and they're true today. And you see, you have to look at the nitrogen content. Importantly, if you look at all the conversion factors you see on the right-hand side of your screen, the conversion factors are not 6.25, they're not 6.38 like we see in milk, but it's actually typically less than 6. So what we're telling the consumers today is not really accurate about digestibility and the quality of protein that's in the product today when it comes from other sources. And Dr. Preston raised this important topic, and he's done some really classic research in terms of when it comes to chitin. But chitin contains a little over 6% non-protein nitrogen, which is well above the level that the FDA considered that was inconsequential. Well, Dr. Preston has some really comments. So Peter, you want to talk about densoviruses?
Well, again, just very quickly, just to highlight the fact that with cricket-based and any insect-based food product, whether it's the quote-unquote cricket flour, whether it's insect flour, whether they are protein bars, what you're doing is concentrating tremendous numbers of insects. And that's not the same as cultures which eat 6, 7, 10, 12, 20 insects for a particular meal, or as grazing in the course of a day. You're getting a tremendous amount of viral content. This is a tremendous inoculum. And these viruses are not the same as they were 200 years ago. They're not the same as they were 2 years ago. So this is why we make such an issue of this. The single-stranded DNA viruses are particularly catastrophic in insect populations, and this is but one example of a tremendous zoonotic reservoir that we are not paying attention to.
I appreciate the remarks. We're all living in this world of within the pandemic, but this is another example. I think we have to be very careful as we transition to look at insects and other sources of protein, because we're not thorough enough. And if we're not thorough enough, we could actually damage public health. So even the EU or EFSA, which the European Food Safety Authority there, made a comment a few years ago, and the point is, it was bolded in this slide: possible presence of biological and chemical contaminants in insect food and feed products. We can't emphasize that more than what we said so far. When it comes to virology, Dr. Pressman, you want to make a few comments, and we'll look at some viruses, particularly infected insects. We have some examples of that. Peter?
This is probably a little redundant, and I think we're getting short on time. So why don't we move ahead here?
These are the viruses to which Peter was referring. So the point here is, you see in the third column, subvertebrate relatives. And the point is, on the fourth column, is that they're common in virtually every species of insects. Does the industry, any part of the industry, look at this? And the answer is clearly no.
Right. I mean, we not only, I think the point here is that we see this variety of viruses, that all of which have been sequenced. And again, we're not running with this data. We're not generating hypotheses to be tested in studies that look at foods and look at what we're considering as future foods. We're seeing the liberation of a whole range of microorganisms, for example, from the melting polar ice caps. What's there? What's really there? Only a tiny fraction of that vast population of very old and novel to our world viruses have been looked at in terms of their genetic stability and their potential pathogenicity. So we're concerned about phytoviral spillover. We're concerned about zoonotic reservoirs. We used to think that it was simply a matter of exotic animals, of camels, of bats. And we've got to widen our focus. We've got to step back, and I think we've got to. We believe we've really got to engage this in the laboratory.
And part of that engagement in the laboratory is what Dr. Pressman actually did in the laboratory. Pete, you want to give a quick run view what this was about?
Well, very quickly, we chose not to throw away the chitin, but we took the exoskeletons from crickets. We extracted, purified it so that we're looking at chitosan, which is a deacetylated version of the chitin, and taking advantage of the multiplicity of amino groups. We're looking at a highly positively charged, we're talking about a tremendously electropositive and linear compound. And what we did, working with Dr. Clemens, we decided that we would attempt to utilize this substance as an interposition graft in simple nerve transections. We took advantage of the fact that most tissue is electronegative. Blood is electronegative. Blood cells are electronegative. Axons are electronegative. So when you look at something that is highly electropositive and has great biocompatibility, is not rejected, does not generate foreign body reactions or other inflammatory reactions, and in effect electrostatically enhances the linear regeneration of transected or hemisected nerve fibers, you've got something that has some real potential. And we used a simple current from a 9-volt battery to create channels within the chitosan scaffolding. And within that scaffolding, those linear channels actually accelerated not only the regeneration of nerve fibers, but the formation of functional synapses as a result of the linearity, we think. So in a very simple insect system, we were able to restore function of limbs that were innervated by these nerves that were experimentally transected. So this all came out as a result of trying to minimize the waste associated with the use of insects for food production.
You could argue that's a part of a sustainability, a function of sustainability for biomedical engineering opportunities.
We hope so.
We were excited about that. Obviously, many of you have seen this kind of a graphic. Companies are changing. This is about a soy-based nut that's no longer made out of chicken. Maybe you are familiar with Beyond Meat practices here. And so it's interesting that internationally, even here domestically in the United States, people are looking for more simplified labels. Well, these are not going to be simplified labels. They're not the quality of protein that one would see. So you see there on the right-hand side the various ingredients that are used for these particular products. Similarly here, we have the Light Life plant-based burgers. The label ingredients are really challenging, and how to make an emulsion system that functions like, tastes like, but is stable like, but it's not the same as, because you have to add a lot of good engineering and good food science, frankly, to put this all together. And part of that protein is that, again, going back to what Dr. Preston was saying a little bit earlier about non-protein nitrogen. And so we took the 4 major protein sources there on the left-hand side, and we did some calculation, look at non-protein nitrogen, limiting amino acids, and look at digestibility. They don't measure up to animal protein. So clearly we have to look at some differences here in terms of managing the quality of protein as opposed to just protein itself. This gives you a microbial approach to fermentation technology. We won't belabor this because of the interest of time. Again, you can have all this material. Algal proteins have been a topic of a number of investigations. Again, they have some challenges with tryptophan and lysine. And then I've worked in the space when I was a diving instructor, and obviously you're suffering with the branched chain amino acids and the sulfur amino acids. These are enough opportunities, but you have to understand that the proteins that are made from these sources are not, in fact, complete proteins. And part of that plant-based protein, we see we want to compare different products that are, these are products that are on the US market. This project though came out of the UK. And you see here, you can't measure up to the quality of protein. There's some variations that you see across the board here. And pick your favorite one, soy, coconut, almond, rice, get that. But the quality of protein doesn't measure up. Matter of fact, that precipitated really important document. I worked in pediatrics for over 20 years. And we see here that even the group there out of the UK said, well, don't feed it to children. These kinds of products are low in calcium, bioavailability of calcium, don't contain vitamin B12, which as you need for a lot of nerve transmission. You also need it to prevent conditions like different types of anemia. So these are not as complete as one would think. As a matter of fact, they're suggesting that maybe rice protein doesn't contain all the polyunsaturated fatty acids, coconut protein doesn't contain all, you see the various minerals that are cited on this graphic, and oat protein, which I actually did a treatise on this maybe a decade ago. The fat content, what you need for growing children, is really not there. So a lot of things you have to consider when you're trying to give this to the general population. Know your population for which you're going to be feeding. Clearly, the graphic here indicates a lot of sourcing protein, and the industry is going to do a good job of looking for alternative sources. But its safety, viability, and allergenicity are really going to be important concepts, as well as the virology of these proteins. And we scanned the literature in terms of the patent office here in the United States. This is what we came up with. So clearly, protein blends are on the way up to give you some satisfaction of some protein complementation to taste and flavor and stability. But importantly, we look at allergenicity issues. This is a database that's public domain. This gives you various sources of non-animal protein, well, animal protein there on the left-hand side, but fungal and plant protein there on the right side. And allergenicity, referring to the potential cross-reactivity, are alive and well. Well, if you look at the plant-based proteins, and this will give you some example, what's the sources of these proteins that are potential allergens, and we've given you them. All the way from asparagus, which we all love, to some of the plants that we have, you see here on the left-hand side. Perhaps a more complete list that you see on the right-hand side, nearly 400 of them. And from nuts to grains, to hemp, to chickpeas, to legumes, they're all there. So we really need to understand these kinds of dynamics for the future. This gives you a nice graphic of what it should look like, the primary allergen proteins as defined by FLAM. And this is again, a really important database for you who are interested in this particular topic. It's interesting that, in fact, the first four, these are the top 4 proteins associated with allergies that are found in plants. Over 2/3 of them contain, 2/3 of the families of plants contain these 4 protein sources. So the source of where we get PR10, parsley, birch pollen, and celery and apples, so we get that. Celery and grains that contain this. And we see here from animals and various forms of bacteria that are innate to plants. And we also see fruits and vegetables. And we continue to tell people eat more fruits and vegetables. We really need to understand the chemistry and the public health implications more than the antioxidants that have been touted. A few more slides here. As we look at allergens, we must include allergenicity. And virtually nobody has done this kind of work. This came out of a friend of mine there in Austria, which is really trying to address this particularly important issue. And we also want to respect the WHO and look at these sustainable development areas, and there are several of them, as you can see. But ultimately, we want to lead the general world in good health. We wish that on everybody, of course. Peter and I have worked in the really challenging areas of the world, and we want to get better health for all of them. We want to, beyond allergenicity, we want to also, in all fairness, look at sustainability. There's not a uniform definition of sustainability. We want to address that. as soon as possible. And they've done a very good job here at the USDA trying to say, what should we address? And you see the 4 areas at the bottom of this slide. In fact, we want to address innovation, science, where does plant fit, and its and various systems. And graphic here, and from the USDA, we want to support better science, the best science, and scientific methods for better approaches to give something better for tomorrow. We want to be effective in what we do. That means a team, international team working for innovation, looking at plant-based proteins and other sources of proteins, of course, and what they might have some impact on the environment. We also want to look at optimize. We want to engage the farmers of the world. And as they invest for future technologies, how do we invest in them to make plant foods more available on a global basis? And importantly here, we also want to look at, is the food system as we know today, is it the very best? So the USDA is investing to look at how do we have better food through better agriculture. So in fact, we get better food to meet the needs of everybody, also be sustainable in terms of working and respecting the land. And particularly, we want to look at water, which is a limiting factor in all these areas. Well, in conclusion, clearly non-meat market is alive and well. But we need to understand not only the market, we need to understand what we're marketing out there. And we need to educate the consumers. Are these plant-based protein or other sources of protein, are they really more healthful? What are the data to support it? And actually, if you look at the literature, there are minimal data to survey that these non-animal proteins are actually more healthful. And are they actually more environmentally friendly, and are they really more sustainable? And what is the role of single cells and extracts and insects for the future? We have to understand those. We're not there yet, obviously. Blends may be the way of a ticket for the future. We want to understand consumer education could be paramount. We could win those who are fearful of technology. And we also look at bottom line is we have to evaluate all these sources for safety, digestibility, and quality. And nobody is doing that except for really fine work at the University of Wageningen. They're really doing some fine work. So if you want to look at another source of this space and protein digestibility and nitrogen, the source, the University of Wageningen has a whole center based on these topics. And we also want to look at analytical chemistry. Analytical chemistry needs to change. So we have a better grasp of what protein is really there, as opposed to non-protein nitrogen. And then what are the health emerging issues? And phytoviruses may be one of those issues. And Dr. Preston and I have done some work in this space, which we can discuss. Bottom line is we may well be in a house of cards. We don't want anybody to fall into the house of cards. We don't want things to collapse. But we do want to be sure that whatever we do, we're providing the best possible help for everybody, and possible help not only for people, but also for planet Earth. And with that, we thank you very much. Thank you very much.
Thank you very much, Roger and Peter, for that absolutely fascinating presentation. You covered so many areas, from phytoviruses to safety implications of protein bars, to protein bioavailability, allergenicity, and alternatives. So there's so much we can ask questions about.
Yes.
So thank you for the questions in the chat. I will come to those in a minute. I'd like to firstly, as chair, ask you one question. How do you see this developing in 20 years? You know, you say we need maybe perhaps more regulation, certainly more safety assessments. At the moment, we're using names where the consumer is sort of a bit hoodwinked, I think, into thinking because things look the same and maybe are functionally the same, like a plant-based burger could be used in the same way as a meat traditional containing burger, for example. What do you think we need to do in terms of labeling and regulation to ensure the population does stay safe, but we also eat in an environmentally friendly way?
Well, you raise an excellent point there, Anita. I appreciate the comment opportunity. Clearly, we need to look at the analytical chemistry and what does it mean physiologically. None of the methods out there really measure physiological response. And I could appreciate the DIAS and the PDCAS methods that have been advanced by WHO many years ago. PDCAS was adopted for most proteins here in the United States, but it's not being used. As a matter of fact, the labeling for that does not even be used in the US. It doesn't require daily values. So they just simply measure kilodal, that's protein 6.25, and that's where it is. Well, that's not true anymore. We've seen that in the various examples that we provided you. I think that internationally requires collaborative efforts to look at protein methodology. And like I said, Jeff Moore and the whole team at USP, US Pharmacopoeia, published a paper, and the FDA took no action on that. So it does require some world action on being truthful and not misleading to consumers. The other part, of course, how do you measure safety? And the safety is more than just microbiological safety. And I think it's look at even functional properties of some of these proteins. And from a food science perspective, they don't behave the same as meat science or meat proteins behave or milk proteins behave. They're markedly different. Solubility characteristics, the configurations are different. And then you can break them down you can partially hydrolyze them. So you get to change the solubility, but you might even change some epitopes. And so the allergenicity might be reduced, but they're not investigating that. I think we have to we have to go back to get the team effort on this particular approach. Peter, comment before Anita has another question.
I think you've covered it brilliantly.
Thank you.
We're passionate about this, Anita. Thank you.
I know, I can tell, and I think we're really feeling that passion, and you know, you're creating a lot of interest here. I've got a question in the chat from Francesca. Is there evidence that pathogen load populations change over the life cycle of insects?
That's a great question.
And nymphs versus adults.
It's a great question. The answer is yes. And I can't, since I'm not a specialist in insect virology, I can't tell you specifics, but I know where to find them. Fanny Balik and Philippe Coulson have done a tremendous amount of work in this area. It's a great question because they do. The pathogen load varies very significantly. The metabolic rate, the nutrient availability, the immunological responses at each phase of development vary significantly and has a tremendous impact on the viral load, the microbial load in general, and again, I can get my hands on those papers, and I'd be happy to send them to you as soon as I do. But it's a great question.
Our entire library is open to Anita, to you, Ming, and the entire Robin, the entire team. So you can have all of our slides, all of our data, and if you can advance it, we're here for you.
And I see while I'm carrying on here, I see that there's another related question here on another really terrific question about the rate of pathogenic disease in places in the world where insect consumption has been high and commonplace, such as in Southeast Asia. And the answer is that it has not been studied. There are I've had a number of conversations with a couple folks who have some expertise in tropical disease at the University of Hawaii Burns School of Medicine and at London at the London School of Tropical Medicine, and there is a real void in this area. And the general response is, well, we don't know. If you don't look for it, and we don't know what to measure, we think it's not there. And we see a tremendous amount of endemic disease in these areas, particularly because it's easy to attribute disease to dirty water legitimately. It's easy to attribute disease to poor hygiene. I've seen a lot of that. I've spent a lot of time in Afghanistan in conditions of extraordinary squalor, and there are diseases there. There are diseases in refugee camps in North Africa that we've never seen before. We're looking at symptoms far more closely now. We're identifying syndromes that have never been described, and we don't know what the etiology is. So the work, all that says is that work that you're asking about really does need to be done, and it has not been done. The sad reality is very similar to to that of drug development, vaccine development for targeting diseases that occur in the developing world, or what we used to call the third world, because there isn't a lot of profit option there, because the market is anemic, forgive the analogy, or forgive the metaphor, that the development was never done. So people are not looking at these areas because there's no funding, there's no sponsorship, and ultimately there's no profit for what could be developed in response. Sad, sad reality, but true.
So thank you for that question, Cassandra. If anyone wants to ask a question by putting their virtual hand up, do feel free. I'm just sort of going through the chat. Saeed, do you want to ask your question?
Yes, I will do, Anita. Thank you. Can you hear me?
Yes, thank you, Saeed.
Yeah, thank you, Peter and Roger. Excellent speech. Many thanks. And I'm glad you highlighted the issue about possible viral infection through insect. My question here, are we really short in food resources? And would it be better to look at the better management of our wasted food in terms of labeling, use by date, where the food still be safe to eat, rather than focusing on insect as a source of protein or source of food, with so many still question mark about the safety of these resources? Thank you.
Oh, Saeed, thank you for bringing that up. We would embrace speaking with you and some of your colleagues, all of your colleagues, and everybody here about how do we better manage food waste, and how can we recycle that. And much of I spent time in Korea and actually a lot of places in the world. Well, recycling food is really going to be helpful in the world, and ensuring safety, particularly for our vulnerable population, is going to be critical. We don't do a good job of, and I actually did this in Canada a number of years ago. We don't do a good job in both in North America about that very important issue that you raised up, Saeed. So we welcome the opportunity to discuss what technology is needed so we could actually take these food products, be sure that they remain safe and feed the rest of the world. Thank you for that.
Thank you.
Yep. You know, this highlights, I think, our frustration about how we even now turn our back on populations that are underserved. And it's remarkable. One of the things that attracted me to a visiting faculty position in the Netherlands Antilles, here I sit on a poor rock in the middle of the Caribbean, was the proximate population, which is terribly underserved. This is very much the third world that I sit in here. I mean, there's a goat on my roof, and there are chickens running wild, and people don't have enough to eat, and half the time they're frankly fried on whatever local stuff that they're intoxicated on. And here I sit in a medical school with a faculty of highly educated people, and we've turned our backs effectively on the indigenous population. It's extraordinary. And this is an example of what happens here. There is a very good medical school here that is tremendously profitable because of the low overhead, of course, and yet we don't follow through with our primary mandate, which is to take care of our fellow human beings. And I think this is one of the things that we've really got to somehow we've got to instill a real discomfort in our students and our colleagues. We've got to provide motivation for rediscovering our service ethic here. I apologize for venting about what's on my mind right now, but I think it's relevant and I think it's very important.
Thank you.
Thank you,
Thank
Peter. Can I say one more thing in addition to what you said, Peter? I'm glad you mentioned all of this, but I would add also that our politicians should work harder. Like I can tell you from experience in the Middle East, hundreds of thousands of hectares of million years old olive tree have been destroyed through this civil war. And you can imagine the negative impact of destroying all these olive tree and field of wheat, cotton.
Yeah.
It is really sad and it's really, really annoying.
Yes, yes.
We share your sadness.
Couldn't agree with you more. Couldn't agree with you
I'm going to, sorry, just wrap it up. Maybe we could have a discussion offline, but I'm just aware everyone has to go. So I'd really like to thank Roger and Peter. I'd like to thank you for your inspiration. I think you've got us all really fired up here in Melbourne. And I want to thank you that you've made us really start asking some really important questions. So thank you for your contribution today. I'd like to thank all the audience members for coming along. I know yet another Zoom is probably not what we all want. It'd be good when we can get back in person, but I really appreciate it. We had a really great audience. I just want to flag that the next event in this series is on the 17th of December. So a bit of a Christmas breakup one, I suppose. I think it's also via Zoom, isn't it, Robin? But we've got Dr. Brad Rideau from CSIRO, who will be presenting his latest research on diets and sustainability from an Australian perspective. So with that, I'd like to thank you very much. Again, thank you, Peter, thank you, Roger, and thank you, everyone who's attended. Okay, thanks, bye.
Thank you, Anita.
Everybody.
Thank you,
Have a good
Mary.
day.
Thanks, guys. It was great. Really appreciate it.